So our first notebook for today is called Digital Representation of Molecules. And notice at the top of my notebook, we've actually got 3 links to the other 3 notebooks here as well. So we're going to help you get around today as best we can. We hope you're able to follow along with everything. And this particular notebook is based on the first 5 notebooks in the Mulsi Chem Informatics course. And I cherry-picked that stuff to pull a few things out that we can use today. And as we go through, the overview for my session is going to be how are molecules represented on computers. I'm going to talk about that briefly. We're going to talk about SMILES, S-M-I-L-E-S, and we'll talk about a few other common molecular formats, file formats we see. And the objective for this is to learn the basics of SMILES and learn to convert molecules from chemical formulas and structures to SMILES. And I should have added this. We're also going to learn how to take a SMILE string and feed it into RDKit and use that. So when we think about using computers, computers do much better with strings of characters than they do with 3-dimensional structures. It's a much faster computational process. So when we work with DNA or RNA, we'll represent that DNA sequence, for example, here by the nucleotides A, T, G, and C. So this is the part of the gene for dihydrofolate reductase from humans. And so that's one way we can represent it as a string. That's the word we use for it in Python. It's just a list of characters. We can also, here's actually the sequence of human dihydrofolate reductase of the amino acid sequence. So each of these letters represents one amino acid. And once again, for a computer to compare strings of characters is much, much quicker than for a computer to try to compare things in 2 or 3 dimensions. I can see a few folks are raising their hands in the room, but there's no access for chatting or Q&A. If you have questions, please go to the Discord server.
Paul?
Yes.
Would you be able to like zoom in a little bit? Maybe that's
okay.
I'm not sure if that's, but I think I would be able to see better. Thank you.
Okay, thanks, Jessica. All right, so as we're moving into cheminformatics, I've got a structure here of aspirin, and it's a relatively simple chemical structure, but for a computer to describe this, then it's going to have positions for all the atoms and all the bonds and types of bonds and so forth. Well, there were 3 very common string conversions for small molecules: SMILES, INCHI, and INCHI-key. We'll be using SMILES, and you can recognize this is the SMILES string for aspirin. We've got some capital letter C's and O's here and some numbers, and I'll explain a little bit about what all of these mean. So this is just going to be a very basic introduction to SMILES. And if you want to learn more, there's actually a link in the notebook. You can read more about SMILES at this tutorial. So click that link and you could go there. I wouldn't encourage you to do it now, but you can go there later. So SMILES stands for Simplified Molecular Input Line Entry System. And so this is the way to convert a 2 or 3D chemical structure into a string, a series of characters. And so if we want to represent a line drawing of ethanol, so here we have, as I explained to my students in general chemistry, when we do line drawings of organic molecules, carbons are implied. So here's a C on the end and a C here. And unless it's shown otherwise, we assume that carbons have 4 single bonds. And so if carbon's going to have 4 single bonds, this one's going to have 3 hydrogens attached to it. The hydrogens are implied. This one's going to have 2 hydrogens attached. Those are also implied. We do show the hydrogens explicitly when they're bound to oxygen. So this is CH3, CH2, OH. Now the SMILES string for this is just CCO. So, and notice these are all capital letters. And so in SMILES strings, we can represent all the atoms as, or all the elements in the periodic table. And if they're 1 letter, then you just use a capital letter. If it's a 2-letter symbol, like thallium would be capital T, lowercase h, things like that. So that's how we represent atoms. We represent bonds. Single bonds are represented by a single dash. But that's implied, and so if you don't put the dash in there, then the program will read it as if there is a dash there. Double bonds are represented by an equal sign. Triple bonds are represented by a pound sign. Aromatic bonds are represented by an asterisk, And if you have two disconnected structures in a single smile string, then that disconnection is represented by a period or a dot. And we have branches in molecules as well, and branches are shown in parentheses. So for example, 2-propanol or isopropanol would have carbon, carbon, and then an oxygen branched from this second carbon, and this carbon is going to be covalently attached to the third carbon. For 2-propanone, we have carbon, carbon with a double bonded oxygen attached to that carbon, and finally a third carbon. So that's how we do branches. This is a very rapid introduction, and we'll have some practice in just a few minutes. We show rings by using numbers. All right, so if we look at the smile string for cyclopentane, if you look at this, starts off with a carbon. We put a 1 after that carbon to say this carbon is going to be connected to another carbon that's labeled carbon 1. So here's carbon, and then there's 3 carbons, and then another carbon 1. That carbon 1 comes back and connects to this first carbon 1. So that gives us cyclopentane. For an aromatic ring, we have lowercase letters. So in pyridine, we've got N1, and then 4 carbons, and then the 5th carbon here is going to be a C1. So C1 is connected to N1 to give us pyridine. So that's a little bit about rings. Here are a couple of examples of smile string, just taking them one atom at a time. So the smile string for 2-propanol, we've already seen this, is CC and O in a parentheses and a C. And so the first C is this carbon atom. The second C is this carbon atom. The O in parentheses is attached to it. And the third C is this carbon atom. And notice where the oxygen, the hydrogen is shown. It's not implied, it's explicit. All right, so that's the smile string for 2-propanol. We get to something a little more complex. This is ethyl acetate, and we're actually going to do the structure from right to left. So the first carbon is shown here, and that lines up with the carbon on the far right in this structure. The next carbon is shown here, so it binds to the first carbon. The third atom here is an oxygen. It's not in a parenthesis, so it's just in the chain, so it is bound to this carbon, and then following that oxygen is another carbon. So this is the 4th carbon that's listed, or the 3rd carbon that's listed there, and there's a branch. The parentheses with the oxygen indicates that there's a branch, and so that branch is this double bonded oxygen, and finally there's the last carbon. So that's ethyl acetate. And notice the ethyl acetate contains an ester group, which is -O-C, open parentheses, equals, capital O, close parentheses, dash. So you could use that for an ester group in any structure you wanted to create in a smile string. Now you can go to a site called PubChem. I'm actually going to go there right now, hoping it will come up. You can go to PubChem for any structure that you want. We'll do this later. And if I want to get the smile string for, well, we've already got the one for aspirin. Let's do caffeine. And when I search for caffeine, the first listing for caffeine comes up. It includes a smile string. So you can find the smile strings there. And so you don't have to memorize smile strings. You can actually find them on PubChem. And every molecule I've looked for has had them. You can actually draw the molecule in the molecule sketcher from the protein data bank. Also, you can draw the molecule and then it'll generate a smile string for you. So we're going to do an exercise here. I'm going to give you about 3 minutes to do this, to write the smile string for ethanol, a smile string for water, a smile string for benzene, and then to tell me which molecule is represented by the smile string capital O equals capital C equals capital O, and then the smile string for an amide group, and then finally the smile string for a 4-membered carbon chain with an amide group on the second carbon. And so what you'll need to do is go in. This is actually an executable cell, but it's all commented. I just wanted you to put the information in here for what you're going to have for a smile string. So with the first question, it's for ethanol. So I'm going to give you about 3 minutes to do that, and then we'll come back and look at the answers. Okay, I'll give you about 30 more seconds. And I'll show you the answers here. And one of the things I do on my exams is I often provide the answer for a later question and an earlier question, and students who catch on to that figure out the answer for this one is actually above. So the smile string for ethanol is CCO. The smile string for water is just oxygen. So we don't write the hydrogens in there, and usually it's not necessary. In some cases, it might be necessary to write the hydrogens in. Smile string for benzene. So this is a ring, and it's aromatic. So it's a lowercase c. And so that's C1. Now we have 4 more carbons, 2, 3, 4, 5. And then the 6th carbon is going to be C1 again. So that gives us a benzene ring. And the molecule that's represented by O equals C equals O, well, that's just carbon dioxide. And finally, the smile string for an amide group, that's going to be a capital C, then a parentheses with an equals O, close parentheses, and a nitrogen. And so that's an amide group. And you can see that I actually provided the answer to the 6th question. And here's an amide group found in that 6th question. And while you were doing your exercise, I looked at the Discord server. I realized that when I did that, I covered up my screen. I apologize for that. But there's a lot of questions there, and there are people answering those questions. So we're going to be talking about smile strings today and using smile strings in all of the notebooks, I believe. But there are a number of different formats that can be used for molecular files. Smiles and INCHI are both strings. The other formats that are shown here, like mol, SDF, PDB, XYZ, and so forth, these are they contain 2D or 3D coordinates. And in fact, we'll be using some of these later because when we do docking, we need to have the hydrogens on the proteins, and we also need to have partial charges on molecules. And so we won't go over that in any more detail here, but we'll get to that in the later notebooks. All right, so the next thing we're going to do is take a look at RDKit. And the question is, what is RDKit? What is an RDKit mole object? And what can I do with RDKit mole objects? And the objective here is to use RDKit to create mole objects in Python. So what is RDKit? RDKit is a library that people started to develop around the year 2000. It was actually developed in the computer language C++, but we have a Python interface to RDKit now. And so that's what we're using. And in fact, that's undergoing development all the time. And so RDKit can create molecule objects. We call them mole, M-O-L objects. And those mole objects can do lots of things. And we'll see quite a few of those things today. And so most of what we can do in RDKit is based on this mole object. All right. And an object is a type of a variable which has specific data and methods associated with it. And so the format we use for calling the data and methods is we have the object name followed by a period, followed by the kind of data we want to get out, or an object followed by a period, followed by the method we want to use, and in parentheses after the method, the arguments. And in most cases, the method needs to be followed by parentheses, even if they're empty. And we'll see examples of that. So if you've worked with Python before, it's easy to create a list. You just say, this is an empty list, my list equals, and then 2 square brackets. And then if I use this command, my list, I'm going to call the method append. And the argument I'm going to have here is a 1. So I'm adding the number 1 to this list. So we're going to do similar things here. So as we go forward, it's going to be more complicated than this with RDKit, but try to keep in mind that we're just calling data or methods from these mole objects. All right. So I also something I meant to mention at the very top, we have two different kinds of cells in these CoLab notebooks, all right? One of the cells is called a markdown cell. So I'm just going to double click on the screen right here, and you can see that there's text here. And the text is formatted here. And so you can use something called Markdown to format text so it appears the way you want. And so that's a text cell. And so CoLab is not going to try to do any calculations in that kind of a cell. The other kind of a cell we have is a code cell. And so this is a code cell. And what we're going to do here is on the left-hand side of the code cell, you'll see there's a play or a run button. So if I hover over this, you can see that the instructions come up to say to run the cell. We can also run the cell by hitting Control Enter. All right. And so what's going to happen, I'm going to run the cell. You should do this too. So hit the run button. While it's running, you get this little dotted line going around the run button. It's going to take it a couple seconds to get oriented. And notice on the left-hand side of the coding cell, we have a check mark. That means it's completed its commands. And we have 0S. That means it took 0 seconds to do that. So once the commands were loaded, the system recognized it was doing calculations. It did it quickly. Now, we're working in Google CoLab. CoLab has lots of Python libraries already installed, but RDKit is not a default installation on CoLab. And so we're going to have to install that. And so we use the command pip install RDKit. In order to do that from a CoLab notebook, we have to have an exclamation point in front of that. And we've already entered all this information. So we're going to install RDKit. And then from the RDKit library, we're going to import the chem sublibrary. So please hit the run button here. And you can see that it's collecting information. It's installing RDKit, and it's successfully installed. Check mark, 6 seconds. So that job is done. We're ready to use RDKit now. So now we're going to create some molecules in RDKit. We're going to use smile strings to do that. So in this cell, we're creating an RDKit mole object. So methane is the name of that object, equals, and chem, so we're calling the chem sublibrary of RDKit. There's a dot there, and after the dot, we have the method mole from smiles. And then there's a parentheses with quotes in there. So we put a capital C in there. So we're going to actually create methane. That the smile string for methane is capital C. So when you hit that run, it created that. Well, did it work? All right, so I'm going to hit the run button here, and we can see that we have CH4. So there's our methane. So we've got another exercise here. What I want you to do now is to create RDKit molecules for each of the following strings. Now, I've actually put the one in for acetic acid already, but go ahead and put in the strings for propane, ethene, cyclohexane, benzene, and I'll give you 3 minutes to do that, and then we'll come back and take a look at that. Okay, we'll give you 1 more minute. All right. Somebody has actually put the smile strings into the Discord server. That's okay. But if you don't look at it yet, if you don't have to, give it a try. about 15 more seconds. We'll start entering some information here. All right. So the smile string for propane is actually given on the right-hand side here. So it's just three, capital C, 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 all capitals. Ethene is two carbons with a double bond, so that's going to be C equals C. Cyclohexane is, there's going to be six carbons, but it's a ring. So we have capital C1, capital C, four of those, and then C1 again. So that gives us, just to check to make sure we've got 1, 2, 3, 4, 5, 6 carbons. Benzene, it's going to be similar, only it's lowercase, C1, C, 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 C1. And then acetic acid is shown here, capital C, then a branch to equals oxygen, equals O, and then close that parentheses, and then a capital O. So I'll hit the run button, and it very quickly created those. It didn't give any errors, and you can visualize the molecule, but just by typing the name of the variable that refers to that RDKit mole object. So if I press run and I have acetic acid in there, I see acetic acid. I put in benzene and hit run. This is funny. I did acetic acid quickly. Took a couple seconds for benzene. All right. And I'm going to do a quick check and see how many participants we have. We have 630 people. Excellent. All right. So what we're going to do now, in this next cell, you don't have to change anything. What we're going to do here, any questions you have on those smile strings that we did, you can put those in the Discord server. In this next cell, we're setting up this notebook to visualize things for us in what's called the IPython console, which is actually part of the draw library, which is part of the chem library. All right. So we're going to import draw from rdkit.chem, and we're also going to import this IPython console and use that. Then we're configuring the way it's going to look. So IPC calls that IPython console library, and it tells it to use SVG to give higher quality images. And then this one is draw options.add atom indices. And so what's going to happen once I execute this is that we're going to get numbers for the atoms. All right. And then this just tells us how big the images are going to be. So I'm going to execute this cell. Now I'm going to go back and execute the previous cell. And what you're going to see is that the atom indices will appear. And they do. All right. So now we're going to do a little bit of working with an RDKit molecule. And what's going to happen is I'm going to go in here, and when I click to the right of acetic acid, so that's a mole object, I'm going to put a period or a dot there. When I do that, I get this whole list of options, okay? And I want one that'll tell me the number of heavy atoms, okay? And so I'm selecting get num heavy atoms. Now, this is a method. I have to put parentheses after it, all right? So what this will do now is that this will give us, we're going to print a statement that tells us how many heavy atoms are found in acetic acid. Now, when we talk about smile strings, heavy atoms refer to anything other than hydrogens, all right? So when I run this, we get the statement that acetic acid has 4 heavy atoms. So the get num heavy atoms method goes in and counts the number of heavy atoms in the acetic acid mole object. Now, if you want to know more about how things work with RDKit mole objects, there's a lot of information in there. And so I'm not going to do it, but if you hit the run button here, you're going to see about 3 or 4 screens worth of information to tell you what goes on with what you can learn or what you can do with an RDKit mole object. All right. What we're going to do now in the next cell is that we're going to take a look at not just the number of heavy atoms, but the total number of atoms. So we want to include the hydrogens. So this is the command we used in the previous cell. X equals acetic acid dot get num atoms in parentheses. All right. And what we're going to do now, Y equals acetic acid dot get num atoms, but we have to put an argument in there so that we will get the hydrogens as well. All right. And the argument we're going to use is only explicit equals false. So I'm going to copy that. I'm going to put it in these parentheses. And I don't need to put quotes around it because these are arguments that are these are recognized as arguments by the get num atoms method. Notice here that false is in a different color. False is a reserved word in Python. You can't make a variable and call it false. Okay, that's a reserved word. Now, we have a print statement here, but we're doing F printing. And F printing, if after the print, the open parentheses, I put an F, then I put quotes, and I can put anything, any string, any text I want inside that quotes. And if I want to include something from a variable, I put it in curly brackets. So I'm going to say there are X heavy atoms, so the number represented by the variable X, and when I hover over it, notice that it says it's 4. There are that many heavy atoms in acetic acid, and Y total atoms in acetic acid. All right. Now it doesn't know that yet because it hasn't run that command. So when I do this, I get the statement there are 4 heavy atoms in acetic acid and 8 total atoms in acetic acid. And I see there's still a regular pattern of questions coming into our Discord server. That's great. Okay, we're going to wrap up by looking a little bit at Pandas and RDKit and how they can work together. And this will be important in the very next part of the workshop. In the very next hour, Pat Walters is going to take you through this. And so we're going to see how we can use Pandas to work with molecules from RDKit and how to visualize relationships between different parts of the data. So the objective here is we're going to review the Pandas data frame briefly. We're going to learn how to visualize structures in a Pandas data frame, and then we're going to learn the apply tool for expanding a Pandas data frame. So Pandas is a library in Python that's used for data analysis and manipulation. And the world of data science, it's all over the place. It's very popular and very powerful. It works. The data frames that it produces are two-dimensional data frames. They have rows and columns. If you need more than 2 dimensions, you need to work with NumPy arrays. So what we're going to do is we're going to import Pandas as PD, and this is just a normal abbreviation that everybody uses for Pandas. And then from rdkit.chem, we're going to import Pandas tools, which is going to enable us to work with Pandas with RDKit. So we can do some RDKit functions in Pandas. And Panda tools, we're issuing the command render images in all data frames. So anytime we have an RDKit mole object, then it's going to actually show up as a chemical structure. All right, say images equal true, so they will appear. Now in the next cell, once again, you don't have to do any change. We need to execute this cell. Almost forgot. All right. And then in the next cell, we're going to use a library called URL-lib. And we're going to use a method from that library called URL retrieve. And from this, we're going to pull a data file that's actually found on the GitHub server. So everybody needs to do this. So hit the run button here. And if it works, you should see this output where it says in single quotes, it says nseds.csv and some other information. And if you try that and it's not working, then you should go to the Discord server and that will help. So what we're going to do now is we're going to load these molecules. So NSAID stands for non-steroidal anti-inflammatory drug. So there's a list, I think there's 21 or 22 NSAIDs in this file. All right. So I'm going to declare a variable NSAIDs file, and I'm going to put the data, or I'm going to put nseds.csv. That CSV file is in this variable. And then I like to, when I do data frames, I like to have the name that tells me what's in there, followed by df that reminds me that this is a data frame. All right. Then I say nseds._df equals pd.read_csv. So that's a method in Pandas that's going to read this file. And when it does, it's going to create a data frame. And then I'm going to ask it to show me the data frame. So this command nseds_df will just display the data frame. So hit the run button there. And you should see the data frame. And this data frame starts with 0 and goes to 21. So it has 22 rows. It has 2 columns. Notice the columns are named there's a name and then a SMILES. And so the names of the NSAIDs are here in the first column, and the SMILES strings are shown in the second column. Now, what we're going to do is we're actually going to add some additional columns here. And with Pandas, we don't have to use for loops to do this. All right. And there's a method called the .apply method. And so what we can do in Pandas, because we've installed the right libraries from RDKit, we can use this to add additional columns to our data frame to contain features of our mole objects. All right. So for example, we can learn the number of letters that are in this column called name. We're not going to do this. I didn't execute this, so we're not going to execute that. But what we're going to do is we're going to introduce another column. So this is a column that doesn't exist yet. So I enter the command, or I enter the name of our data frame, NSAIDs_df, and then in square brackets, and then inside quotes, I have molecule. And then I'm going to call the column that contains the SMILES strings from our data frame, and I'm going to have .apply, and then I use chem.mole from SMILES. Now in this approach, we don't put the parentheses after mole from SMILES like we did before. It's not required. And so what I'm going to do is I'm going to run this cell. So go ahead and run this cell. When you run this cell, you'll see the first, now notice it's NSAIDsdf.head. So that's just going to give us the first 5 rows. And so it gives us the first 5 rows of this data frame. And so we get the name, the SMILES string, and the molecule. And notice that the atoms in the molecule are numbered because we turned on numbering earlier. So that's one look at that. I'm going to go a little bit further here. So this one now has 3 columns in it. We're going to add 4 more columns because these are drug molecules. And there are things that we want to know about drug or drug-like molecules. And so from rdkit.chem, we're going to import something called descriptors, one library called descriptors and another one called Lipinski. And the descriptors is going to enable us to get the molecular weight and the log of the partition coefficient for the molecule between water and octanol. And then the Lipinski library enables us to get the number of hydrogen bond donors. It's called NHOH count, and the method for hydrogen bond acceptors in Lipinski is NO count. And then we'll take a look at the head of that one again. And so now we have our data frame is getting bigger. So we've added 4 additional columns just from this previous cell. And so you can see the molecular weight is shown in this column, log P in the next column, the number of hydrogen bond donors, and the number of hydrogen bond acceptors are then shown. And I'm going to show you 2 more tricks you can do doing a time check. Okay, this will take about 3 minutes, then you'll still have 15 minutes to catch up and take a break. So what we can do from data frames is there's some plotting that's built into the data frames. And so what we're going to do now is I'm actually going to have the data frame call a histogram plotting function, and it's going to plot a number of the data parts that we have here. So it's going to give us a plot for the molecular weight, the log P value, the hydrogen bond donor number, hydrogen bond acceptor number. It's going to give us a histogram for each of those for all 20 items on this list. So when I run that, it's going to take a couple seconds. And so now we have a histogram that tells us how many of the molecules in our data frame. There are 2 that have molecular weights that fall into the 150 bin and so forth. The most popular bin is the 250 bin, and there are 4 elements there. Log P values are shown here, and you can see that there are this many elements in that, or this many items in our data frame that fit to that have that log P value. And the same, we have the distribution of hydrogen bond donors and hydrogen bond acceptors. So that's, and once again, this is a plot that's built into Pandas data frames. Another one that we're going to use, and you'll see variations on this as you go through the day, is we're going to actually do a bar plot of the molecular weights of each of the drugs. All right, so if you hit the run button here, you're going to see a plot, and in this plot, on the x-axis, we have the name of each of the drugs that's shown here. On the y-axis, we have the molecular weight. And then we have plot here, the bar graph represents a molecular weight of each of these compounds. So salicylic acid has a molecular weight, I think we saw 138.12 before, and it fits right there. And now notice this is not a highly deeply labeled or annotated plot. I'm not sure if it's possible to do that from within Pandas, but this gives you a quick look at your data, which is really what you want to do with plotting from Pandas.